I'd like to say good evening because it's evening here, but I realize most of you it's morning, so whatever time, good day. As was said in the introduction, I'm going to talk about the use of LabKey at Nestlé and particular attention to the assay and the study modules. So I've been asked to give a few words about the institution that I work for, the company that I work for. I'm assuming that most of you have heard of Nestlé before, so I'm actually going to use this little moment to discuss the science that has been done at Nestlé over many years. So Nestlé is a company that has a rich history of innovation, starting back in the 1860s when Henri Nestlé produced one of the first specific infant foods, infant formulas, if you will. And it goes all the way to very modern things like what we released last year, which is a fully vegan cheeseburger analog that has your analog of meat, bacon, and cheese, all in a vegan manner. So all of this, of course, is founded on science and on research. And so Nestlé has a very developed R&D organization. And this R&D organization goes from basic research to produce discoveries through the development and the deployment in factories of new production mechanisms, of new products, and so on. And I myself work at Nestlé Research, which is here on the left, firmly in the discovery area. So this means that of this entire chain, we are the people who are maybe most similar to academic researchers in that we do science to a degree for science's sake, for understanding. And due to that, we're kind of a particular organization. There are about 900 people working at Nestlé Research, 50 different nationalities. We're in 4 different geographic locations. I myself am in Lausanne, Switzerland, and we produce a certain number of publications and patents and so on every year. So we tend to be a little bit like the academic people in that we want to publish as much as possible. We want to get knowledge out there in addition to what we're delivering to the company. And in so doing, we have a lot of collaborations with various partners, but also with a lot of universities. And this leads to particular way of working and a particular way of working specifically around data, the way of exchanging data that people in the academic communities are used to, a lot of ad hoc exchanges of data, a lot of different data formats, and so on. So this is the context within which we're using LabKey at Nestlé. So I'm going to talk today about 2 challenges that we are facing, that we are addressing through LabKey. The first of those challenges really is the organization of omics data as such. So to illustrate this, I'm going to take this simplified chain of events in any life science project or experiment. Generally, when we do life sciences, whether it's in a cell culture dish or in a clinical trial, we collect a sample from a living entity. So this can be harvested cells, or this can be a biopsy, or a urine sample, a blood sample from a clinical trial, and so on. There's a lot that happens in the lab on this sample. We create different aliquots, we transform these samples, extract DNA, purify proteins, and so on, in order to be able to perform measurements. And these measurements then give us some form of raw data. So typically, we have a mass spectrometer, it will give us mass spectra. We have a sequencer, it will give us raw reads. And this raw data needs to be processed in some way, and the results need to be reported in such a manner that they speak to the biological problem we're trying to address, the question we're trying to address, and are amenable to our data analytics systems and people. And so coming into this, we had a certain number of elements already in place. So we had a system that was responsible for all of the physical world tracking, basically. So we have a LIMS system. It tracks our samples, it tracks our transformations, it tracks everything that we do in order to prepare our measurements. but we didn't really have a solution for the management of the data, all of this data processing, the reporting of results, and this is where LabKey really comes in. And the complexity that we were trying to address was that it's easy to have a single system for the LIMS part because everybody collects samples, everybody does lab work on them, prepares them in some way to put them in an auto sampler or a 96 well plate or something to put them on the instrument to measure. But the data processing is a bit of a different beast because we have every technology that has its own software, its own systems, its own mechanisms to process the data and its own specificities, what type of information needs to be carried along. Much of this is file-based, much of this is vendor-specific software that cannot necessarily be automated or integrated into automatic workflows. But in the end, we really need all these results in one place where they can be collected by the data analysts and used. Additional complexity comes from the fact that, as I stated earlier, we have a very large number of collaborators, and they also send us lab data that that needs to flow into this data flow in a constructive manner so that it can be combined with the rest. As a kind of a contrast to the previous talk, we don't currently put clinical variables, all the patient information and so on, into LabKey. We have other systems for that. So we're maybe a little bit upstream of what was shown earlier in that what we're looking at is people who are running analytical platforms who are collecting data from samples, processing that data and reporting the results. But in the end, we're striving for the same thing, and that is to have the results in a structured manner within the platform, within LabKey, so that it can be collected from there and either shipped to other analytical systems or directly analyzed based on LabKey itself. So how have we solved this problem in LabKey? So LabKey allows us to access files that are stored within the file root, the project level file root of a project. So what we have is basically a large file storage system which all of these platforms place their raw data on, and then if they have their own specific software, vendor software and so on to analyze this, they will process the data using their existing systems and produce results that can go into LabKey. So the solution for the file level as a first approach was simply to connect LabKey to our existing file storage system so that the files become available in LabKey and can be imported into LabKey. And then we performed a certain amount of optimizations to allow the various different platforms to easily import the data that they were already creating through import scripts. And we leverage the automatic synchronization that we have with our LIMS system to pull in all of the sample information. And this allows our platforms to basically work with generic sample identifiers. They have sample barcodes. They don't need to know that this sample comes from a subject in a clinical trial with a certain visit associated to it. They only need to know this is the identifier of their sample. And LabKey will then take care of pulling in the information it needs from our sample data. So I'm going to switch over to my browser now and give you a quick demo of what I'm talking about here. So this is our staging server. None of this data is actually live data, but it's to illustrate basically what I've just discussed. So this is a demo study, and within the demo study, we have our file storage, and this file storage, as I said, is accessible in other ways to the various platforms, and they can place their data there. And so we're going to go into the metabolomics platform here, and I don't have all the upstream part, but we have an Excel sheet in this case that has been created by this platform. And this Excel sheet is in a format that is specific to the metabolomics platform that they have been working with in the past. And we have an import script that allows us to import the metabolomics data. And this is where some of the mechanics, the technical aspects that we've put in place come into play. This is as you would see it if you have never used LabKey before. Once this is set, this becomes a default setting for that user, and they can actually just click next, and get their data in. But in the interest of illustrating what we've done here, I'm going to go in and do this configuration now and say that the participant and visit information for the data I'm uploading comes from an existing list. And so I go in here and say it's a list, and the list that I need is the sample mapping list. And this will allow LabKey to associate the barcodes and the unique identifiers of the samples in the Excel sheet that's being reported on directly into the LabKey tables. Here I'm just going to click on Finish. I'm not going to put any comments in. And as you can see, this turns into a background job that runs. And of course, there is an error because I'm doing a demo. Okay. So I'm not gonna redo this here now, but the idea is basically that we then go into the assay module, and in the assay module we have a table that contains the unique identifier of the sample that was used to upload the data. We have the participant in the clinical trial, or the sample that was the properties of the sample that are important associated with it, as well as the time points for the visit. And this all becomes available directly through the combination of the data from the samples and from the unique identifier directly in the assay. And this assay can then be uploaded into the study module in the way that you saw in the previous presentation, to be able to explore that data and combine it with data from other platforms within the same project. I'm just going to jump back to my PowerPoint and move on to the second aspect of the talk today, the second problem that we're trying to address, and this is data provenance. So we often find ourselves in a situation where we have a document with a set of results, a set of insights. This may be an internal report. This may be a paper that's been published. This may be a patent that's been submitted. But what we'd really like from a data quality point of view, from a good practice point of view, is to be able to determine where all of these results came from that are being reported here. So what did we actually use to produce this figure, and what were all the intermediate steps? And this can be relatively complex. So we have a bunch of samples that were collected. We have all our sample tracking that is that meets all the quality standards, but all of this element in between of obtaining raw files, processing them in a specific way, combining them together, integrating them with other data, is something we also require tracking on that was not in place prior to our use of LabKey. And this is where the new Provenance module comes into play. So what we required and what we discussed with LabKey was to have the ability to record provenance between arbitrary elements within LabKey. So we have samples that are present in LabKey, we have files that are present, we have tables in the assay module, in the study module, we may have analysis reports and so on. And we'd like to have a connectivity between all of these in order to be able to trace back exactly what happened. And we'd like these connections to be annotated with the necessary information to understand what happened. So which versions of software were used, what reference databases were used, and so on. And we'd like this to be automatable in the sense that any action that is performed directly in LabKey is automatically recorded. And we'd also like to have API endpoints so that when we do have automated data processing, this automated data processing can be connected up with LabKey and can provide LabKey with the information to say that this processed file comes from this raw file, and that such and such software was used in such and such version with such and such database, and so on. So I'm going to jump back to my demo here. And I'm going to go into the proteomics data here and show, in this case, how this would be manually recorded. It's clear that the manual recording is a certain amount of overhead, and wherever possible, we would want this to happen through APIs. But the manual recording allows me to show what it is we're recording and what kind of data on provenance is then recorded within LabKey. So I'm going to take these three raw files, and I'm going to create a run. And I'm going to say these are input files. And this gives me the run builder. So the run builder is part of this provenance module. And I'm going to put in here that this is my demo run, just to give it a description. And I already have the inputs. I'm just going to add a file quickly here and say this is my output. So I now have inputs, I have files, I have outputs that are files. I could also have samples that are inputs or outputs. And I have properties here. So in the properties, I can add various properties that are important for this step that is being recorded. And I'm going to say this was an Orbitrap instrument. I'm using the human database to recognize the various peptides in my spectra. And I'm going to say this is the 2020 October version of the database. And then I click on save here, and it will save this run. And if I go to the experiment module, I will be able to see this run, this demo run here, as if it was an experiment. And if I click on it, I can view it as a graph. And I can view it in a new version of the viewer in which I have the context of these three files ran through this demo run into this, this output file. And potentially this chain can then be extended to have a large tree structure or graph structure with samples at the top, raw files, semi-processed files, processed files, and then go into the table of the assay module to show this is the origin of that tabular data, and then from there go even further and say there's an R report that was created from this, and the R report used a query over certain different tables, so that we can have the full audit report, if you will, of how a certain result was obtained. So yeah, those are the two aspects I wanted to show, to discuss and demo today. Clearly, there's still a lot of work to do. The provenance module is shaping up nicely. It's ready to complete the task we want to complete with it. The manual run builder is there. The APIs are there, but it's now in our camp here at Nestlé to continue on with the work and integrate this with all of our automated data processing solutions. The visualization of the provenance graph that I showed is still very much a work in progress, and we'll continue to develop together with LabKey the improvements on that side. Ontologies were already discussed. They're also a big subject independently of what I've shown today for us here at Nestlé. So we're very interested in seeing the evolution and the elements that were shown today in the roadmap on that front. And the study module is a module that is very much oriented towards human clinical trials or animal experiments, in that it has a participant, it has certain recording of time, whether it's continuous or visit-based, but it doesn't necessarily meet all our needs for cell-based experimentation, for recording of cell-based experiments. So we're running various options through our minds at the moment to see how we can leverage LabKey to really record that in a structured way as well, and build that data into an asset within Nestlé. So with that, I'd like to thank you all for your attention. Thank all the people at LabKey who have been instrumental in getting us to the point where we are today. And yeah, I'm ready to answer any questions you may have.
Oh, we do have a question. Question is the Provenance module open to everyone?
So it is a feature in the enterprise version of LabKey, and it's still under development. So it comes with a little warning saying it's not yet a solid API, a static thing. It's still going through frequent modifications, but it's available for use in the enterprise version.
Great.
Yeah, and this is Adam. I'll just add that our plan is to make that available to all of our enterprise clients in 2021, once we've kind of stabilized it and established those APIs.